Good evening. My name is Dennis Berkey, and as provost of Boston University, it's my honor to welcome you to the Boston University's 59th University Lecture. I want to say how good it is to see such a full house, and especially to see so many students here this evening. You are all very welcome. The University Lecture was established at Boston University in 1950 for the purpose of honoring members of the faculty engaged in outstanding research. The lecture provides an opportunity for all members of the university community, as well as the general public, to hear a distinguished scholar discuss his or her area of expertise. Early each year, all members of the faculty are invited to submit nomination for the subsequent year's lecturer. The 5 previous university lecturers act as the nominating committee. It is customary that a brief question and answer period follows the lecture, and we shall observe that custom this evening. This year's university lecturer, Dr. Charles DeLisi, is a scientist of profound imagination and transformative intellect. Cited for his distinguished scientific leadership and achievements by President Clinton, by then U.S. Department of Energy Secretary Elliot Richardson, and by the Smithsonian, Dr. DeLisi recognized early on the value to human health and to the environment of a reference human genome sequence. Acting on this vision while serving as a director at the U.S. Department of Energy, he mustered the scientific and political support necessary to launch what came to be known as the Human Genome Project. Today, as generations of scientists and engineers advance, harvest, and apply the fruits of this initiative, Dr. DeLisi continues to press the envelope of its implications, standing, as Newton, on the shoulders of giants, looking across centuries, not just decades, to foresee wonders such as humans influencing our own biological evolution and even speciation. The considerable glamour and practical potential of this work does not detract Dr. DeLisi, ever the model scientist, teacher, and entrepreneur, from his steady focus on his primary concerns: the cell as a system of interacting proteins and genes, a persistent global perspective, and a deep concern for the ethical and policy implications of the rapid technological advances in the biomedical sciences. Charles DeLisi was educated in the public schools and the City College of New York. He completed a PhD in physics at New York University, a postdoctoral appointment in chemistry at Yale, and an appointment as staff scientist at Los Alamos National Laboratory. His developing interest in biology then took him on to the National Institutes of Health, where he was appointed founding chief of the section on theoretical immunology and then head of mathematical biology. In the 1980s, he and his collaborators were the first to identify protein motifs and used them as rapid predictors of genetic function. Path-breaking work on T-cell antigenicity and T-cell receptor recognition soon followed. In 1985, Dr. DeLisi was appointed director of the Health and Environmental Research Programs at the United States Department of Energy, where he founded the Human Genome Project. In 1987, he was appointed professor and chairman of the Department of Biomathematical Sciences at Mount Sinai School of Medicine in New York. Dr. DeLisi was recruited to Boston University as Dean of our College of Engineering and Professor of Biomedical Engineering in 1990. During the following decade, he literally transformed that college into a research-intensive center for the productive synthesis of engineering, scientific, and medical knowledge and discovery, with particular emphasis on the development of our nationally recognized programs in biomedical engineering. He did this work the old-fashioned way, engaging each faculty member in an ongoing discussion of his or her interests and ideas, the best and most effective kind of mentoring a university can hope to support. In 1998, Dr. DeLisi was appointed the university's first Arthur G.B. Metcalf Professor of Science and Engineering. Since completing his term as dean, Dr. DeLisi has continued to serve as the director of the university's Center for Advanced Genomic Technology and the founding director of the nation's largest graduate program in bioinformatics. He is our senior associate provost for bioscience. Charles DeLisi has authored or co-authored more than 200 research papers in leading scientific journals. He holds 5 patents, and he is co-founder of 2 biotech startups, Pharmadyne Incorporated, concerned with AIDS and other viral diseases, and Boston Array Technologies Incorporated, concerned with proteomics. As we consider the increasingly broad scale of what we look to research universities to do, from educating undergraduate students to achieving the breakthroughs that will cure diseases and propel society's advance, Charles DeLisi stands as a most worthy and inspiring exemplar. We are delighted to present him to deliver Boston University's 2003 University Lecture entitled Crossing the Watershed, Biological and Other Worlds in the Post-Genomic Era. Dr. Charles DeLisi.
Thank you. Thank you. The 20th century began with a major scientific revolution, which paved the way for perhaps even more important technological revolution toward the end of the century. In the former case, I'm referring to the revolution in physics, particularly in quantum physics, and in the latter case, computer technology, computer science, especially information technologies, and the computational science that it makes possible. Like most transformative technologies, and perhaps even more than the printing press, computer science is having major ramifications, one of which is that it spawns new technologies. And one of those technologies is biotechnology. And in fact, that revolution is going to impact not just the way we see ourselves biologically, but it's going to impact our understanding of the origin of the human species, the origin of all life on this planet, human health, agriculture, and inevitably, it will impact technologies which traditionally have very little relation to biology, especially engineering, where the relationship has been in the past engineering assisting the development of biology, but not the reciprocal. What you'll see in the future is that we'll develop whole new engineering technologies, and that's part of the reason I think people from the faculty of the College of Engineering are interested in this, whole new engineering technologies that have never before been thought about. So what is it? What is the human genome? What is the genome? The genome has the ring of the commonplace. It's the complete collection of genes of an organism. And a gene, if I can have the first slide, is simply a very large molecule. When you abstract away all the details, it's simply a sequence of chemical bases labeled here A, T, G, and C. And that sequence, just as a sequence of letters in a word, that sequence contains information. The information it contains is the information for a protein. It's basically the blueprint for the production of a protein. Proteins are the biological machinery, the things that make cells operate, the hemoglobin that transports oxygen to our tissues, the antibodies that protect us from disease, the collagen that gives substance to our tissues, the myosin that assists our muscles, and so on. But the genome is much more than its chemistry. And that can be seen immediately by realizing that in the fertilized egg of a human being, the human genome, in a 2-cell embryo, something small enough to fit in a volume less than 1/1,000,000th of an inch, has the information for the spontaneous formation of a human being, the spontaneous formation of the heart, the liver, the central nervous system, the lungs, basically the biological development of a human. The program for understanding that development will occupy biologists for most of this century. But once it's understood, that is bound to have a big impact on engineering and on manufacturing engineering in particular, because it's the way nature has solved the problem of manufacturing something that's very, very complex. Basically, we know something could be done by looking at biology. We know there's a solution. We know that it's possible to place in a very, very small volume, in some convenient code, the information for the spontaneous development of something very, very complicated. That we know, we just don't know how to do it right now, but we will learn how to do that over the next 50 years. There are other implications to the human genome, in particular, that engineers should be interested in. One of the big engineering problems is to understand complex adaptive systems. An example of a complex adaptive system is the 747. It's certainly complex enough, and basically if you put it on autopilot, it will adapt within certain limits to the environment. But that is a very, very primitive complex adaptive system compared to human cells. And here what I've done is presented a cartoon which is supposed to represent part of a lymphocyte. This is blown up about 10 million times. This is a cell that's supposed to respond, that takes part, produces antibodies, takes part in protecting us from environmental agents. And what is shown here is foreign substance. Think of it as a small molecule. It could be a toxin, any contaminant. The cell has to be able to recognize foreign substances that are nearby, and it has to be able to distinguish them from all the self-components, from millions of other self-components. And when it does that, when it recognizes those foreign components, it has to transmit that information to the nucleus, where it activates certain sets of genes. In particular, it has to activate the genes for antibodies and other mediators of the immune response. It also has to activate other genes which enable the transport of those molecules, of those mediators of the immune response, out into the environment of the cell where it can neutralize the invasion. It has to activate other sets of genes that reproduce the cell. That is, the cell becomes an armamentarium for the production of agents that neutralize an invasion, and it also activates genes for the reproduction of those factories. And it has to do this while talking to other cells in its environment because it needs help. It doesn't do it alone. So it has allies. And while it's doing this, it has to modulate the environment to see whether, in fact, its response is effective. And if it is effective, it has to turn itself off in an orderly manner. But it doesn't turn itself off completely. It doesn't revert to its initial state. It leaves the factories there to some extent. It leaves the assembly lines there. It stops munitions production, so that it's able to respond very, very quickly to any subsequent invasion. Now, understanding that, we're a long way from understanding that, but where we are right now is in a position to understand that process in a collective manner. That is to say, in the past, this is a process which involves thousands of molecules, and in the past, biologists were only able to understand one molecule, study one molecule at a time, or perhaps two molecules at a time. We're now, because of the genomic revolution, in a position to understand processes that involve thousands of molecules at a time, and we can foresee a day in the not-too-distant future when we'll be able to understand complex adaptive systems of this sort, and that will inevitably impact engineering and all kinds of things in society. Now, like all important technologies, all important areas of science, the impacts of genomics, the ramifications will go deep. We've had two conferences in the past 6 months, one sponsored by the Pardee Center, in which we discussed the future of human nature, and another one last month by the Philosophy and History of Science, in which we discussed some of the ethical implications of genomics, the near-term implications, which involve civil liberties, and perhaps the longer-term implications, which involve reprogramming, redesigning the human genome itself. And I'm not going to be talking about any of that tonight. Tonight's presentation is going to be strictly scientific, basically an epithalamion between genomics, biology, and computer science. So those two examples are how I think genomics will impact two of the ways in which genomics will impact technology, but we learn more than that. By looking at the human genome, we also peer deeply into evolution, the evolution of life on the planet and the evolution of our own species. And what this shows, this lower branch here, where it says 12 to 16 million years, shows the Asian orangutan branching off from the African apes. And as we move up the tree, 6 to 8 million years, we see the gorilla branching off from humans, chimpanzees, and bonobos. And you go up further to 4 to 6 million years, where you see the closest common ancestor of those three. What's significant about this diagram is that humans don't have their own branch, as was previously thought. Humans, from the point of view of DNA, humans are African apes. And actually, every time I say that, I think of the debate between Bishop Wilberforce and Thomas Henry Huxley 150 years ago, in which Bishop Wilberforce begged to know if it was from his mother's side of the family or his father's that Mr. Huxley claimed to be descended from the apes. And actually, Huxley won that debate, although sometimes I wonder. After this 4 to 6 million years, in which our genus, Homo, branches off, there are other speciation events, the most recent of which occurred about 130 to 150,000 years ago, very, very recent, in which modern humans emerged from an archaic sapiens. And that will have some bearing on some remarks I'll make shortly in a little while. Well, it was questions of this sort, basically curiosity about life, curiosity about its origins, that drove the genomic revolution. But in fact, it was medical genetics that paved the road, and paved it with gelt. What this slide shows is a portion of a hemoglobin molecule. This is a molecule which transports oxygen to the tissues. And it shows a defect. That's this triplet of letters here, these bases, GAG. And there's a single misspelling of this, and it's a single misspelling. It's this single misspelling that's responsible in large part for sickle cell anemia. I mentioned this disease because it was first understood over 50 years ago. And in fact, the story of it was understood fairly clearly by Linus Pauling prior to Watson Crick. Pauling wouldn't have had anything like this in front of him. This is in the late 40s. Linus Pauling was on Vannevar Bush's Presidential Advisory Committee for the Future of Medical Research. And on that committee was also the Dean of Harvard Medical School. And they were having a discussion, and the Dean of Harvard Medical School mentioned that it was the arterial blood that was affected in people with sickle cell anemia, which Pauling didn't know. And Pauling, as soon as he heard that, said, "I understood the disease." And what he understood is what happens is a small portion of the hemoglobin molecule, which is normally water-soluble, that portion, becomes water-insoluble. And what happens is you now have something which you could think of as a little drop of oil in water. And when you place a drop of oil in water, the oil sticks together. It doesn't mix. It's not soluble. And that's what happens. Now you have hemoglobin molecules, and they have these little patches of oil on their surface, and they're not going to want to mix with the water, so they mix with each other. The result is, and you could show this thermodynamically as well, the hemoglobin starts aggregating. It distorts the cell. It buries oxygen sites to some extent. Cells become fragile, arteries occlude, and you have all the manifestations of the disease. The disease, we now understand, is a little bit more complicated than that, but it was a fairly clear understanding. In fact, Pauling was able to suggest a diagnosis on the basis of that. But after more than 50 years, there's still no cure for sickle cell anemia. So that is something worth keeping in mind as we hear about all the promised medical miracles, which will come, but for different reasons, I think. The next slide shows something of a different nature. We now fast forward about 40 years, and at this point, in the late 80s, computers have started to emerge as a tool in biology. When the gene that's involved in sickle cell anemia was sequenced in the late 80s, the first thing people did is they asked the computer whether, in fact, anything like it had been seen before. There's a very famous algorithm called the SmithWaterman Algorithm, named after Temple Smith and Michael Waterman, and Temple Smith is on the faculty here at Boston University. That algorithm probed the memory banks of a computer the way Smith and Waterman themselves don't understand, and the answer was yes, we've seen something like it before, and we have some idea of what the function was. And the function, the idea that came back was that this has something to do with a molecule that transports ions from outside of a cell to inside a cell. In fact, that's what it does. It transports chloride ions. And again, in this example, a single defect in this whole molecule, which is very, very large, it's much larger than hemoglobin, a single defect is responsible for cystic fibrosis. You have to inherit the allele from both parents. I'll say something about that a little bit later. But again, it's very rare. Now, the next slide, we start to get into situations where we have a genetic disease which is not rare and not the result of a rare mutation. And this is late-onset Alzheimer's disease. And in this case, again, there's a single misspelling, 897 letters, one misspelling. That's this T to C here. This base T becomes a C. And what happens, a variation like that. Now, if you inherit, let me backtrack a bit because this is a little bit more complicated. This is not the result of a rare mutation. It's the result of the fact that genes have alleles, and in this case, there are, because we evolved so recently, there are typically 3 or 4 common alleles per gene. The alleles are different versions of the gene. Think of the gene as a word, and think of an allele as a synonym for the word. So each of these words come with 3 or 4 variants, 3 or 4 alleles, 3 or 4 synonyms on average. And in this particular case, there is an allele. One of the alleles is not quite as synonymous as the other 2. And if you inherit that version from both parents, the chances are 45 to 50% that you'll develop late onset, you'll develop Alzheimer's disease late in life. And in an audience this size, there are maybe 10 people who are at risk for that. Whereas with cystic fibrosis, the probability is that there's not a single person here, probably maybe 10%, 0.1, that there's at most one person here that would have cystic fibrosis. So there's a big difference in the commonality. And here, this arises from the very nature of human heterogeneity. It's not a mutation. It's just it's a combination. It's the wrong allele. Now, most common disorders have more than one gene involved. And just to illustrate the origin of some of the complexity, well, what this slide shows is heterogeneity, but it's the wrong type. This shows diversity in the population, but it's misleading because it's the wrong type of diversity. Most variation, in fact, is between groups and not within particular ethnic groups. It's far more productive to focus on variation, susceptibility, and resistance to disease. And to understand the magnitude of that problem and the extent of variation across the population, just look at a particular case. And here's an instance of 5 genes, let's say, and each gene has 3 versions, 3 synonyms, 3 alleles. And think of these, for example, as genes that might be involved in defense against something. And maybe they're proteins that repair mistakes in cells, or maybe they're components of the immune system. There are many, many such genes, but here we're only talking about 5 of them. Each has 3 alleles. And so how many combinations can you have? Well, here's an example of a person, for example, which has allele 1 of gene 1, allele 2 of gene 2, allele 2 of gene 3, allele 3 of gene 4, and allele 2 of gene 5. That's one particular individual. There are something like 243 paths through this matrix, 243 combinations of this sort, and for each combination there's a slight difference in effectiveness, so that these alleles are not all equally effective, just the way synonyms are not all equally effective, depending upon the context of the sentence in which they're used. And depending upon context here, alleles have different effectiveness. So different combinations of alleles have different effectiveness in terms of, for example, protecting in this case. Actually, this is an oversimplification because I haven't considered the fact that each chromosome may have different alleles. And if you do that, this number of 243 becomes more than 7,000. So we have 7,000 variants here of one trait, or an oversimplified trait. 7,000 variants. Basically, you have a continuum of responses with the environment. People in the same environment, 7,000 possibilities. In fact, the human genome has an immense number of possibilities. If there are 30,000 human genes and each has 3 states, you've got 3 to the 30,000th. That's like about 10 to the 13,000th power. And even if you take into account that genes are not inherited independently of one another, that number is immense. To give you an example, there are chemists usually studying in a volume of a liter of water, there are probably about 10 to the 25th molecules. That's a 1 with 25 zeros. Here we're talking about a 1 with 13,000 zeros. So variation in the human population is extraordinary, and even if we consider a few individual traits, we have enormous variability and enormous variability in the way we interact in the same environment. Well, how do you go about characterizing that? Back in the mid-'80s, a number of us thought that having a reference human genome would be an important step in allowing us to approach the problem of human diversity. And now, at first thought, that sounds a little bit strange, because what a reference would do is tell us about those properties of the genome that are invariant across the species. It would tell us how many genes we all have. It would tell us which gene is on which chromosome, which gene is next to which gene, and so on. Whereas we want diversity. We want to understand diversity, not commonality. Well, the key is that any 2 particular genomes differ by about 1 base in 1,000, 1 letter in 1,000. That means if you have a reference genome and you want to determine a region of any other genome, all you'd have to do is find that 1 base in 1,000 difference, and you'd have the other genome. Tremendous reduction in work. Now, back in the mid-'80s, in fact, the technologies were not available for doing that, although there were emerging technologies that we could see. Chemists were developing types of mass spectrometry, which weighs these objects. And in fact, the one that emerged that no one foresaw, and I certainly didn't foresee either the technology or the person that would be the major person behind the application and advance of that technology, and that's Charles Cantor, who's here in the audience tonight. And what the type of mass spectrometry that he's developed does is it actually weighs, it actually determines differences by weighing these molecules. Now, it has to weigh something that weighs about 1 billion trillionth of a pound. In fact, not only has to weigh it, but has to determine differences in weight. That's quite a trick, but chemists have learned how to do it. So the genome was sequenced. Technologies have become available for understanding, for rapidly finding differences between different genomes. And we knew from the very beginning that it wasn't just the human genome that was going to be affected, but the genomes of many, many other organisms. And in particular, one of the biggest beneficiaries has been understanding information about microbial genomes. And here's just to summarize, there are 143 now fully sequenced, fully characterized, completely read microbial genomes, more than that. And there are more than another 500 in progress. And some of the examples of fully read infectious agents are Plasmodium falciparum, H. pylori. Now, H. pylori is significant because that's responsible for ulcers. 10 years ago, people thought ulcers were a lifestyle disease. They didn't realize it was infectious. Same thing with chlamydia pneumonia. That may be, in fact, that may be an infectious agent implicated in coronary artery disease. So infectious diseases, I think, even though genetics drove this, I think infectious diseases will emerge as something that will be a major beneficiary, understanding them and their etiology and what we might be able to do about them. And I'll say something about that in a little while with another entirely different infectious disease, namely HIV. Now, the other aspect of this, of having all these genomes available, is kind of interesting. At the beginning of the project, people were very worried that we're going to have this tremendous amount of data and we wouldn't be able to say anything about all these genes, all these sequences. We wouldn't have the foggiest notion about what they do. And part of the reason for that was that the SmithWaterman algorithm, which was the method of choice and still is the method of choice, in order to learn anything from it, you had to have examples of the genes in some other organism. And since we were going to have all this new information, we weren't going to have examples for most of it. Well, interestingly enough, the very fact that we have all this information now has allowed us to develop entirely new approaches to understanding function. And that has been a major development that resulted simply because of the volume of information we have. The information itself allows the development of new methods. And in fact, it allows the development of not just new computational methods, but new experimental methods, one of the most significant of which is illustrated here. This is a schematic. This is a microscope slide, what you see with all these little dots here. It's about the size of it. And each little dot is a probe for a gene. And this little microscope slide can detect whether any number of genes are on or off. So basically, you can get the signature of a cell. You can determine which genes are on and which genes are off in a particular environment, and you could probe 10,000 at a time. So we've gone from studying one at a time to thousands at a time, which is what we have to do to understand disease. Now, one of the clear implications of this is the enormous potential for diagnostics. Avi Spira, who was then a pulmonary fellow 2 years ago, working with one of our PhD students in bioinformatics, Adnan Derdi, looked at the difference between lung cancer, adenocarcinomas of the lung, in frankly cancerous cells and precancerous cells, and found that there were about 50 genes that were either overexpressed, that is much more active than they ought to have been, or much less active than they ought to have been in cancer cells relative to the precancerous cells. Now that immediately suggests the possibility of a diagnostic. If you could show that difference between precancerous and normal, you'd have something very, very important. But we don't want to do just diagnostics. We'd like to identify targets. That is, we'd like to treat the disease as well. And to do that, using this method, you might think, well, we've already learned there are 50 genes or so that are overactive in a cancer cell, and why can't we target those? And the answer is that most of those genes are effects of the disease, not cause of the disease. And if we want to target something, some of them look like if we target them, they might be helpful. But in many cases, we have no idea what most of these genes that are over or underexpressed, over or underactive, do. No idea, meaning we don't know the context in which they operate. It's like having isolated words and out of the context of a sentence. And unless you understand the context, you don't really understand the meaning. So here's an example now. What this shows, think of these little circles as genes, and it shows the sort of information we're beginning to get. And the lines between them show correlations between the genes, think of them as proteins, in fact. And the lines between them show correlations between the way proteins that function together work. So we're in a position of having, for example, a text written in a language we don't know, and we begin looking at it, and we begin to see that certain occurrences of words that we don't know tend to occur together with high frequency, and certain triplets so we see occur with high frequency, and certain quadruplets occur with high frequency. And if you get enough information like that, you can perhaps begin to understand the language, and that's the position we're in. We know, in fact, a lot about those correlations in bacterial systems. And one of the things we can do in the particular system I was just talking about, the adenocarcinoma system, which occurs in humans, we don't know anything about the context of those genes, or very few of them, the context in which they're over or underexpressed, what else they interact with, what they're correlated with. But we know a lot about bacterial systems. So in fact, what we could do is something a little bit more sophisticated than what was done in the past with SmithWaterman. We can look at those genes that have, for example, that are similar, that have the same genes in yeast, different versions of the gene, but very, very similar in yeast, and we can look to see what we know about the way they function in yeast, what their context is. That's what's done here. You look at what the context is in yeast, you then eliminate all genes that are in human, and go back, and what you have is a hypothesis about a human network. So this basically shows a hypothetical network in a human cell, in a human cancerous cell. And it immediately suggests experiments. In this particular case, the interesting molecule is that there's a gene which is involved in vascularization. Tumors actually develop under low oxygen conditions, and they have to vascularize and transport oxygen, and there are certain genes that are overactive in tumors, and this identifies one. and it immediately suggests a hypothesis for what to look for next, and I won't get into that in any detail. But the point is here that we're able to study thousands or 10,000s of genes simultaneously, which is what we have to do because the biological processes are very, very complicated and involve multiple genes. What I want to do now, in the short remaining time we have left, is go on and discuss infectious disease, and AIDS in particular. And now we have another dimension of complexity because we have two interacting populations, a human population and a viral population. And it's a story of virus infecting a human responding, a virus adapting to the response, the human immune system adapting to that adaptation, and so on. So what happens when a virus infects a human? It winds up in the bloodstream, but it doesn't linger around in the bloodstream waiting to be eliminated by antibodies. It infects a cell, and once it's inside the cell, it inserts itself into the DNA, and it can't be seen by the immune system. But the immune system can still tell the cell is infected. It can't be seen by antibodies, but the immune system can still tell, and the reason it can tell, if we go back and think of what a normal cell does in the normal course of things, a normal cell is synthesizing proteins, and it's degrading those proteins. Fragments of the proteins that are degraded are transported to the surface and presented there, bound to a human molecule called a major histocompatibility antigen. So we've got this complex of a fragment of a human protein and a large human molecule which is holding that protein. Here, this is MHA. In this case, it's holding a viral protein. Well, the same thing happens. So we have a cell, a normal cell, and it'll have hundreds of thousands of these complexes, MHA, peptide complexes, fragment complexes. And in a normal cell that's not infected, it'll all be normal. So in fact, that won't be seen by the immune system. The immune system has learned to ignore that. But you've got cells with hundreds of thousands of these objects sitting on them. When the cell is infected with a virus, every once in a while, some small fraction of those complexes hold viral fragments. So we've got basically a chimera, a human-virus hybrid, a chimeric monster sitting there on the surface, and perhaps 1 in 1,000 of these complexes is abnormal like that, and very few cells have them, but the immune system is still able to recognize them. So it recognizes it, and when it recognizes it, and that's what's being shown here, this is a cell of the immune system, a cytotoxic T cell, which is recognizing this chimeric monster sitting on the surface of an infected cell. And when it recognizes it, it goes to work on it with utter ferociousness. It literally explodes the cell, it blows it up, and then it chews it up. In 200 million years of evolution, it's learned not to fool around. But even while this is happening, in fact, the virus is in other cells and it's mutating away. And over time, some combination of viral mutations and the fact that some of these infected cells go into sites that are not easily accessible to immune cells and control mechanisms on the immune response itself, and some combination of those all conspire to give the virus an advantage over time, and after maybe a decade or more, the immune system finally collapses, and when it does it fairly precipitously, and the person is in big trouble because there's no more immune system to protect it. And what's vicious about these viruses is they affect the very cells that protect an individual. They affect cells of the immune system. So what to do about this? Well, the key is mutation. And what has to be done is what has to be targeted. The virus can mutate. It mutates very readily, but it's not free to mutate all over the place. It has to survive. And there are certain parts of it that can't mutate without itself being damaged. That is, without putting the virus in a state where it could no longer function. So there are certain parts of it which we say are conserved. They cannot mutate, or they cannot mutate very readily. So what you want to do is you want to find whether, in fact, there must be such regions, and you want to identify them. Well, how do you identify them? The way you identify them is by sequencing. What you sequence now are multiple variants of the virus. And for HIV, fortunately, there's been a lot of genomes, a lot of variant genomes that have already been sequenced. There's probably 1,000 of them. And what you do is you compare those thousands of sequences. Now, here's a segment of a particular protein, and these little letters stand for amino acids of the protein. We don't have to worry about what they are, but this is a segment, and this will go down the page, and there'll be 1,000 such segments of this sort, and they're all aligned, and you look to see whether there are any segments which remain the same. And here, this region in blue This is to indicate that this little sequence, this little segment, remains the same in all the virus strains. And when you find those are potential targets, because it means in the 30 or so years where this thing has been around, it's been around longer than that, but when it's been visible for over 20 years, all the mutations that have occurred, almost none of them have occurred in this particular segment. So for decades, in fact, the virus has had a very, very hard time mutating this segment. The segment is highly conserved. So the idea is that segment, when bound to an MHA molecule, is the target, is what you want to target, because although many of these complexes, many of these chimeras mutate like crazy, in fact, those chimeras that contain these particular segments that don't vary, because if they did vary, the virus would be in trouble, those are what's targeted. Targeting, finding what to target, what to deliver a drug to, what a drug has to recognize, is really the difficult problem. There are many ways to develop drugs once you've identified what to target. So these are potential targets. And this strategy, this is a rather different strategy than the one that's typically used these days. That's been used for over a decade now. This is a strategy based on immunology as opposed to a strategy based upon knowing the structure of a particular molecule, which is what most drugs against AIDS are directed at. So this strategy will work, in fact, on any agent that infects a cell, and mostly viruses, but there are other things like tuberculosis, which also infect cells. And it will work to identify, and it's particularly important for those agents that mutate readily. And I mention it because it's an example of another application of sequencing. The sequencing in this case is trivial because viruses are small, and to sequence 1,000 variants doesn't take much time at all. But the fact is that a generation ago, you could never even have considered anything like this. It would never have been thought about because we weren't in a position to sequence at all, let alone 1,000 strains of something. So where are we? Let me just review some of the things I've said. There have been multiple impacts of genomic science. The impacts I haven't discussed are the cultural impacts. What genomes do is tell us something about our biology, but in fact, how we approach some of the social issues might tell us more about ourselves than the genome does directly, as the genome tells us very little about human psychology, but I think we'll learn a lot about ourselves psychologically and sociologically by how we respond to some of the challenges, and maybe we can discuss some of those challenges in a little while. With respect to the biology, the impact on medicine will be enormous, and impact on biology, on evolutionary biology, on developmental biology, on engineering will all be enormous. Forensics, I didn't mention, and here I have, this is all forensics, it's all evidentiary, but here I have legal forensics in mind, and I think DNA technology has made an enormous contribution to human justice by showing us which people were unjustly accused. Remember the words of Genesis 18. Abraham comes close and says, "Would you really sweep away the innocent along with the guilty? Heaven forbid for you to do the thing like this, to deal death to the innocent along with the guilty." Actually, those words tend to have a New Yorkese construction to them for some reason. You know, a lot of famous people come from New York, and so it's not surprising Abraham might have been one of them. Well, as you recall, Abraham managed to convince God that if there were 50 innocent people in Sodom and Gomorrah, he would not destroy the cities. And then in what is perhaps one of the most outrageous pieces of chutzpah in the Bible, he argued God down to 10 people. Now, the response of molecular biology to Abraham's treatment of God is not 50, not 45, not 40, not 30, not 20, not 10. Not a single innocent will be swept away with the guilty. Such is the evidentiary power of DNA technology. And with that, I will say thank you, Dennis. Thank you, David. Thank you, Christopher, Jackie. Thank you all.
Thank you, Charles. You were brilliant. I hope you'll take a few questions and comments from the audience. There are microphones in each of the aisles. Please come to them.
Yes.
Charles, well done, brilliant and philosophical as well. Knowing that you were involved with the Genome Project in the early days, in a whimsical moment, I figured that crafty guy that you are, you would have had your own genome sequenced. But I'll ask the cocktail party question. Whose genome is it that we have sequenced? Now, I assume that it is not one.
There are two.
But I assume that it is a club. And so how big is the club? And do they know who they are? Is it still possible to get into the club? How did the early adventurers at DOE and NIH go about deciding who would be in the club? And in the end, does it matter?
Well, I don't think it matters a great deal. There are two genomes, quote, reference genomes, one public and one Celera, and Charles Cantor probably knows the details, and I think one of them does involve a single individual. I think it's the Celera one, and I think we can guess who that is. And the public one, I think, is a gamish of probably many people. Is that right, Charles? And so, in fact, I didn't try to decide. In fact, Walter Gilbert once accused me of not having thought things through because I didn't decide whose genome was going to be sequenced. And my response was, who cares? You know, it's a reference genome, and it doesn't have to be anybody's in particular. And my standard answer to that question when people would ask me, well, whose genome will you sequence? After all, there's 6 billion sequences in the world, and well, how could it possibly mean anything? Because everybody's genome is different. So what are you going to do with the genome? And of course, it's the idea of the reference that really mattered, and I didn't really think it mattered all that much as long as it was a well-chosen. We could have chosen the wrong reference, obviously, but I don't think Craig's reference is too bad. Charles, you have anything to add on that? It's okay.
Well, to what extent did you have to decide that you need to have 100 or you need to have 500 in order for it to be represented?
Well, that's a different, I mean, the question of how now we're in a position where we might want to do things like build a catalog of common variants, and then you need some number of well-chosen people, if you wanted to do that, whose genomes you would sequence, and 50, that's a statistical, that could be calculated statistically. How many genomes, if you choose them properly, how many genomes would you need? Remember, there are 3 or 4 alleles per gene, roughly. So if you want to get all the common alleles, there are many alleles for each gene, but there are 3 or 4 common ones. And if you want to get all of them, you'd have to sequence enough genomes, and not very many. I did the calculation once. It's about 50 or so, 50, 60, to get maybe 90%.
Hi. You mentioned that there's an enormous diversity of different human genomes, suggesting that there's enormous variability among human beings. On the other hand, we also hear a lot from the popular media that our species has less genetic diversity than other species. Right. And we also hear, so I'd like to hear a little bit about that, about what that means for how to conceive of ourselves as human beings. And we also hear recently that we share so many of our genes with other species, and some numbers were floated around this last year. We're similar. Are we similar to a banana? Are we similar to so on? So.
Well, less diverse because we've been around not quite as long. We've only been around for a few million years. So not as long as a chimpanzee, so we expect to see less diversity because there's been less time to diversify. Well, we, modern humans, have been around only 140,000 years, so there can't be very much diversity. That's why there are only 3 or 4 common alleles per gene. But there is great similarity between us and the chimps, between us and mouse. I have a poem that I quoted in the back of the written part, basically to celebrate his written takeoff on To a Mouse by Robert Burns, written by John Updike, in fact, commenting on the similarity between the human genome and the mouse genome. So there is a lot of similarity. It's probably less than 1 base in 100 for us and chimpanzees. It's very, very close. Now, when the chimpanzee is going to be fully sequenced, I think that's going to be a monumental when we have that information. We'll understand a lot, including we should be able to begin to understand why monkeys don't get AIDS, why chimps don't get AIDS, and we do. So, yes, you have a question? Yes, yeah. Who's next? Yeah.
Charles.
Oh, hi, Boris.
So increasingly, science is no longer a gentleman's game, like it was in the beginning of the century, where science was a thing of the very few educated. And the Human Genome Project is a perfect example of how you have to explain the need for great projects to the populace. And I'm a graduate student in bioinformatics, and I always hear these kind of doomsday scenarios where the computational biology has, exhausted itself and so on and so forth. And I guess what I want you to comment about is as you get into projects which are increasingly complex and the explanations for which are increasingly complex, how do you justify these things to the general public? And what do you promise them for their sacrifice, for their investment? And yeah.
But there's a famous physicist who was in front of a congressional committee once and arguing in favor of the superconductor super collider, and he was asked by congressmen, what is this going to do for our quality of life? And basically what he said is it's not going to do anything for our quality of life, but it's going to make us worthwhile as people. It's going to do the sorts of things that make us human, that differentiate us. It's going to enable us to continue to understand the world about us, which I think is almost an organic part of being human. It's very fundamental to our psychological nature. In terms of but more to the point, in terms of the Human Genome Project, there's a tremendous educational problem. I mean, if you want to explain, there are going to be enormous implications for genetic disease, for example. And trying to assimilate that information, understand what the quality of it is, what it really holds out the promise for, what it does do, what it does say, what it doesn't say, I think that's very, very difficult to understand and very complicated. And I think it's going to take a lot of education to explain to people what the limits are and to explain to people when we do have the sequence and everybody's sequence is on a little chip like this, that little chip is not you, it's not an individual. In fact, it was Lewis Thomas, I think, who said that the genome alone specifies only a vertebrate vegetable. It really isn't the essence of what it means to be human, but it is important to human biology and is important to human medicine. But understanding how to interpret the results and how to explain what options people have is, in fact, if people are presented with when they're young, with you're susceptible to this whole range of diseases, and what action do you take on the basis of that information? Well, that information is very complicated. Just susceptibility has limits. There are probabilities associated with it. We're in situations now where we don't know, where you can diagnose, but you can't treat, really, or cure. And do you want to know under those circumstances? And if you do want to know, if you do want that information, how do you protect it so that other people don't have access to it, or the wrong people don't have access to it, your employer, your insurance company, and so on? There are laws now are not, property laws are not what they ought to be. There are no states which say the DNA is personal property. So there are enormous issues of this sort which involve education and involve legal issues, which I think we need to grapple with, and we need to do it much more quickly than it's being done, I suspect.
So I have a quick follow-up. If you were a young man in science
I am a young man.
Sorry. What project would you be advocating for the great next investment in science, and why?
Well, I think we're at a point where we can, not everybody, most scientists will not agree with this, particularly molecular biologists, but I think we're at a point where we can really begin to understand physiological systems. We're talking here about a cell, and it's very complicated. And in a certain sense, it seems maybe ludicrous. We can't even understand the cell as a system to scale up and start talking about organs. But you know the way you do modeling, there are certain things you parameterize. You don't need to understand. Indeed, you can't understand every level of detail. You can't get to the next level of detail by understanding every component to the present level. But there are ways to understand the next level up the hierarchy. I think, in short, that we're in a position to understand a lot about organ-level physiology, and we can do something very similar to what has been done in genomics, basically. And there are people, Herb Voigt's in the audience here, in biomedical engineering that are doing just that, the auditory system, that are pulling together all the information that's available about the cochlea and other aspects of the ear from all over the world, setting up large databases, developing models, biophysical models and signal processing models so you can play tunes to the ear and they can be interpreted. So we're in a position to understand organ by organ and how organs couple and to make real progress there. But that's a huge information technology problem, and it's also a huge social problem, just like the human genome, which I thought was never going to happen when I was at NIH and thinking about it. I thought it would never happen because it meant enormous cultural change in the way things are done. And that cultural change happened. And a component of that change is not just the cultural change among the biologists, but the need for computer scientists and that community. I think it's a real story in the sociology of science to see the dexterity with which the computer science community responded to the genomic challenge. That all had to happen. The problem was a problem. In retrospect, it doesn't look like it was a problem, but it's certainly a change that had to happen before such a project could be possible, and it did happen.
Thank you.
Yes?
When you first set out to achieve this goal, what kind of moral and ethical guidelines did you keep in mind while you were pursuing this project, and what kind of guidelines do you think should be kept in mind for the future so that this knowledge won't be exploited?
Yeah, while I was still in Washington, in fact, before I left, about 3 months before I left, I put in place, in the midst of great controversy of the staff there, I put in place a component of this which said that we'd spend 3 to 5% of the budget for genomics on ethical and legal studies. And that was approved, and I left 3 months later and didn't watch what was going on. And what went on is the person who succeeded me eliminated that. I didn't think it was a good way to spend money. Fortunately, Jim Watson arrived at NIH a couple of years later and started this spending 3 to 5% on ethical studies. It's really the first time, in the past, and for example, with nuclear energy, things were done retroactively. I mean, in the case of nuclear energy and the war, there was an emergency situation, and I think even though there were physicists who were thinking deeply about it, like Leo Szilard, there was nothing formal happened, and we weren't in a position then to put in place the controls that needed to be put in place, and we're still suffering from that 40 years later. In the case of genomics, in fact, we're trying to do things in parallel, and there have been, over the past 15 years, there has been a lot of very, very good thinking that's gone into understanding the complexities, the legal and ethical complexities, and the moral complexities. It shouldn't just be legal, but it's morality that's involved, and how we treat problems in the short-term problems of privacy, of human dignity, of civil liberties, and in the longer-term the possibilities for, in fact, polarizing society economically even more than it is now polarized, because these technologies, some of them will be very expensive, particularly the reproductive technologies. The reproductive technologies, which is where there'll be a major impact in 20 years, the costs aren't necessarily going to come down because they involve specialists. They involve highly skilled people to carry them out. It's not like computer science where the cost is coming down. And those technologies could have a very, very big impact on the next generation, producing generations of very healthy people for those people who can afford the technologies, and not so healthy people for those people who cannot afford, and you get further polarization in society. So those are major, very, very serious problems that have been thought about, but there's been very little action. There's been very, as I said before, and of course we have George Annis here in the audience who could speak to this with great authority. There's been very little progress. There's been some progress in terms of government insurance, but there's been very little progress in general legally, and varies from state to state. Progress in privacy is really very, very fragmented. There's more, you have more protection on your Blockbuster card than you do with respect to your human genome. You've got privacy with Blockbuster. Nobody is entitled to know what you've rented.
Hi, I have a question for you about one of the examples you mentioned. You discussed sickle cell anemia, and you have to forgive me, I'm just beginning my biology career, but as I understand it, sickle cell anemia, part of it has to do with an adaptation for malaria, and I was wondering and I'm speaking strictly scientifically because I understand this has more implications as well, but how does some of the research you're doing or fixes for diseases such as sickle cell anemia, how do they affect the process of natural selection, and that possibly changing parts of the sickle cell anemia could inflict problems on the idea that it was an adaptation for a disease?
It depends on how you're trying to bring about cure. If you're trying to do it through genetic engineering, then there are potential problems that you might muck around with things, particularly genes that are inherited. It depends on whether you're doing germline or somatic cell changes. But in the case I know more about, which is the case of HIV, we're not talking about genetic engineering. You just want to kill the cells. In the case of malaria, the same thing. You want to kill the infection. You don't need to do genetic engineering to do that. That's one way to do it. And when you do genetic engineering, you wouldn't do it on germline cells. You do it on somatic cells. So whatever changes you make wouldn't be passed on to the next generation.
Why don't you take this last question here?
Yes.
Hi. My question is do you see in the next 50 years a technological leap that will change the way genomic sciences works? Like, in other words, is there a bottleneck in technology right now with computer technology coming to a halt, kind of? Do you see some kind of a leap in the next 20 years or so? Do you expect such a thing?
You know, it's always difficult to extrapolate because every year what happens is if you look at progress, and every year we do 20 years worth of work based in the past. If you look at the way computers are evolving, for example, or you look at the rate at which sequencing is occurring, if it's occurring exponentially, what that means is that next year if it's doubling, it means next year you've done everything that was done in the preceding 20 years, if things started 20 years ago. So it's very difficult. In the case of computers, and the estimates I've seen by Hans Moravec, for example, indicate that in 25 years, there'll be computers that will have the processing capability of the human brain for $1,000. And in 50 years, there'll be computers for $1,000 that will have the processing capabilities of every brain on the planet. Now, that doesn't mean these things will be conscious, although at some point I think that'll happen, but it does mean, for example, that it'll process information at the same rate, visual information, auditory information, so that you can easily imagine a computer 25 years from now flying a high-speed jet by itself, or certainly cars being driven by themselves, because visual information will be three-dimensional visual complex information will be processed at the same rate that we currently process it. So it's very difficult when you have capabilities of that sort. It's very, very difficult, and then tying them in with technological developments and scientific developments in other areas to say exactly what will happen when. Beyond that, we can only talk about what might happen on the basis of laws, physical laws that we know about right now, or technologies that we know about right now. And typically a new discovery might take 25, 30 years to make an impact on society. So in many ways, the best you can do is extrapolate out 30 years if you want to do it in a concrete way based upon what we currently know. So I really can't say too much beyond that. I think no one could say very much beyond that. We can say there's certain things that will happen, but when they will happen, I don't really know.
We see the strategy is emerging now to get all of those human-like computers to enroll at Boston University and continue the advance of this marvelous man's work. Charles, thank you very much for a wonderful... You've been a great audience. Our program is concluded. We'll see you next year at this time.